So welcome everyone and Happy New Year. So this is Environmental Research Seminar and Webinar series, and I am Sungkyun Park, a faculty member of Epidemiology and EHS. So this seminar series is sponsored by the Michigan Life Stage Environmental Exposures and Disease Center, M-LEEaD. So today's speaker is Dr. Viveka Thangaraj. Dr. Thangaraj is a postdoctoral fellow in the lab of Dr. Vasantha Padmanabhan in the Department of Pediatric-Endocrinology here at University of Michigan. She received her PhD in Genomics and Oncology from the University of Madras in India. Her research centers around the developmental programming effects of prenatal exposure to environmental chemicals on long-term metabolic health of the offspring using omic tool. So, let's stop. Let me stop here and then. So, Viveka, yeah, why don't you begin?
Hello, everyone. Thank you for that very nice introduction, Dr. Park. So I'm excited to share my research here in this platform. So let me go into my topic, which is preconceptional and gestational exposure of sheep to a real-life environmental chemical mixture alters maternal metabolic profile in a sex-specific manner. So before I start, I would like to acknowledge my PI, Dr. Vasantha Padmanabhan, for providing me the facilities and the expertise in this analysis of this work. Dr. Maureen Kachman from the Metabolomics Core for the metabolomics analysis. My collaborators from the UK, Michelle Bellingham, Neil Evans, Kevin Sinclair, and Richard Leah, who are primarily working on the handling the animal samples. I also thank my lab mates, Dr. Nadia Sadat and Dr. Catherine Halloran, for their valuable inputs in analysis and writing up of this work. I also thank the funding agency, which is NIEHS. So all of us are exposed to several environmental chemicals on a day-to-day basis. And many of these environmental chemicals are also categorized as endocrine disrupting chemicals, as they mimic endocrine hormones and bind to their receptors, resulting in disruption of various biological processes. Some of these examples of endocrine disrupting chemicals include BPA, phthalates, DDT, DDE, atrazine, and PCB. So we are exposed to these chemicals through the food, contaminated food, through the containers, plastic containers that we use, through personal care products and other exposure routes. Along with the exponential increase in the use of these chemicals in recent time, we also find an increase in the incidence of non-communicable diseases, which include cancer, cardiovascular diseases, and diabetes mellitus, with these diseases contributing to 70% of deaths globally. So although exposure to these chemicals on a daily basis is harmful at any day, there are critical windows of exposure where the body is most susceptible to the adverse effects of these chemicals. So these phases include the prenatal phase, early childhood, and the puberty phase. There are several research, animal and epidemiological studies which have shown that exposure to EDCs affects the maternal fetal milieu, leading to adverse pregnancy and birth outcomes, both of which are contributing factors for adult onset of these non-communicable diseases. And although a lot of research has been made with respect to single exposure studies, the real life scenario is not a single exposure because we are exposed to a mixture of all of these chemicals at different doses, and it's a chronic exposure. Although in recent times, there's a lot of focus on mixtures research, where we combine several groups of these environmental mixtures, as these are known to have additive, synergistic, or combinatorial effect, but we do not have an exact knowledge of the outcome of these effect of these mixtures. So in this scenario, we have used a real-life exposure model, which employs the use of biosolids. So biosolids are nothing but treated human waste, and hence they represent both the dosage and the array of chemicals that humans are exposed to on a daily basis. So sheep are exposed to pastures treated with these biosolids, which represent a unique model because this is a real life representation of the low dose and the mixtures of chemicals that we are exposed to on a daily basis, and hence offers a realistic assessment of risk to human health. So the use of a large animal model like sheep, which is a precautial model, which is similar to the which reflects the human developmental trajectory, is also an advantage of this study. So prior studies using this model have already established that maternal exposure to biosolids results in these adverse effects in the offspring. This includes increased bone mineral density, disrupted fetal reproductive and neuroendocrine axis, spermatogenic abnormalities, mixed testicular atrophy, altered ovarian proteome, and the genes involved in ovarian development. And there's also been reports of increase in thyroid weight. So a lot of these effects that we see are known to be sex specific because studies have shown that the environmental chemical exposure effect varies between the sexes, with many studies pointing towards a male fetus being more susceptible to these studies. So why did we choose to study metabolomics? Because it has been shown that the maternal metabolic intrauterine environment has an important role to play in the development of the fetus, and it is also an important metabolic programming agent. So circulating metabolites like pyruvates, amino acids, and lipids are known to be mediators of maternal fetal communications. So the levels of these metabolites often communicate the needs of a developing fetus to the maternal environment, which results in physiological effects including increased intrauterine blood flow or increased glucose metabolism depending on the needs of the fetus. So hence, we believe that this metabolome of the maternal animals represent a crucial link between the exposure to biosolids and the effects that we see in the offspring of these animals. So several untargeted metabolomic studies have been instrumental in identifying both biomarkers of exposure and also the effects of these biomarkers by showing the endogenous metabolites that vary in response to these exposure studies. So with this background, we had the hypothesis that exposure to biosolids perturbs the maternal metabolic milieu, which is consistent with the adverse phenotypic outcomes that's been observed in this model. We also hypothesized that fetal sex-based differences exist in the maternal metabolomic response to the biosolids. So easy care use were maintained on pastures treated with biosolids, which we refer to BTP, or with inorganic fertilizers, which are the controls. And these were exposed from 30 days prior to conception throughout the gestation period. So blood was collected at day 90, which represents the mid-gestation period from same-sex pregnancies. And these samples were analyzed using mass spectrometry platform in both the negative and positive ionization modes. The metabolites were identified by matching the retention time, mass, and isotope profile to authentic standards. Univariate and multivariate statistical analysis using the peak intensity values were done using the MetaboAnalyst software. So the untargeted metabolomic analysis identified nearly 2,500 features in the negative ion mode and 3,815 features in the positive ion mode. As you can see, most of these features were unknown, that is, they have not been annotated using prior standards or experiments. So we only chose the annotated metabolites, which is 1,006 from the negative ion mode and 1,295 from the positive ion mode for our analysis. Outliers were removed, and we were able to get 105 metabolites which were significant by the t-test and 203 metabolites which were significant by the VIP plot. So we combined the differential metabolites from the negative and the positive ion modes. So we used a unsupervised principal component analysis and a supervised OPLSTA analysis to look at the overall metabolites in the negative and positive ion modes. we can see a clear difference between the control and the biosolid groups in both the negative and positive ion modes. So while the PCA shows a moderate separation, the supervised OP-TA shows a clear separation between the two groups. And this is reflected by very good R²Y and Q² values, which is a representation of the goodness of fit and the reliability of these models. So this indicates that there are significant differences in the metabolite profiles of the control and the biosolid groups. So the variable importance in projection scores. So these scores reflect the importance of these metabolites to the variability seen between the two groups. So we combine the VIP scores and the t-test to identify the total differential metabolites. The tables here reflect the top 10 differential metabolites identified from the negative ion mode and the positive ion mode. So we wanted to look at what are the different classes that the differential metabolites represent. And interestingly, we found nearly 40% of the differential metabolites belong to the lipid and lipid derivative subclass, followed by the organic acid and derivatives groups. Taking a closer look at the different species level contribution of the lipids, we could see that a majority of them were fatty acyl lipids, followed by glycerolipids, prenolipids, and other lipid subclasses. Among the organic acid and derivatives groups, nearly 90% of them were amino acids and peptides, which are major building blocks of the developing fetus. We then did a pathway analysis to identify which are the pathways that are disrupted by these differential metabolites. Here again, we saw a lot of amino acid pathways like valine, leucine, isoleucine biosynthesis pathway, amino acid tRNA biosynthesis pathway, and the phenylalanine, tyrosine, and tryptophan biosynthesis pathways to be the most affected pathways. So taking a closer look at each of the subclass of the differential metabolites, we could see that the different class of lipids showed a difference in their profiles in the control and the biosolid groups, while fatty acids, prenolipids, and sphingolipids showed a decrease in the biosolid groups. The glycerophospholipids and glycerolipids subgroup of metabolites were increased in the biosolid groups. Among the other groups, organic acids were decreased, nucleotides were increased, alkaloids were increased, and phenyl polyketides and benzenoids were increased in the biosolid groups. So because lipids are comprises many subclasses, with each of them having separate biological functions, we wanted to take a closer look at each of these subgroups of lipids. So as you can see, the fatty acyl subgroup of lipids was the most represented group in the differential metabolites. So the fatty acyl group in itself has different subgroups. So one group is the fatty acid subgroup, which play an important role in maintaining the plasma membrane dynamics, and they also act as a source of energy for the developing fetus. These are also precursors for bioactive compounds, and all this implies the importance of these class of metabolites in the development of the fetus. So here we see an accumulation of long-chain fatty acids in plasma indicated by the higher levels in the biosolids group, but some of these fatty acids are also lower in the biosolid groups. So the low reserves of fatty acids in the mother could be reflective of increased transfer to the fetus or low reserves in the mother. So this scenario reflects an impaired fatty acid metabolism, which is known to affect oocyte development and spermatogenesis, which has been observed in the biosolids offspring phenotype. The other class of fatty acyl is the acyl carnitins, which are involved in the transfer of the long-chain fatty acids into the mitochondria for beta oxidation, which provides energy for the growing fetus. So lower level of acyl carnitin means less transport of these fatty acids to the mitochondria, which ultimately leads to impaired beta oxidation, which could be impairing the fetal growth as well. Among the other classification of fatty acids are the fatty amides, which are bioactive endogenous signaling molecules. So oleamide and palmitamide, which are both part of the endocannabinoid receptor system. So the endocannabinoid receptors have pleiotropic roles with respect to fetal development. and they are involved in oocyte development and maturation, regulation of bone formation, and also regulation of the reproductive neuroendocrine axis. So the other fatty amide here, which is decreased in biosolid group, is an intermediate in the branch chain amino acid degradation, which we'll see in detail in future, in the subsequently. So the other fatty lipid groups are the glycerolipid groups and glycerophospholipids, which are both increased in biosolid groups. So these are essential components of the membrane, and they maintain the membrane dynamics. Increase in phospholipids are also associated with decrease in methylation of genes that are involved in fetal development. So sphingolipids, which are active membrane lipids and which are critical for fetal growth and bone remodeling, were decreased in the biosolids group. So taken together, the profile of these differential metabolites indicate a clear disruption in the fatty acid oxidation and also increased lipid storage. And both of these are characteristic of chronic oxidative stress. So moving forward to the other major class of differential metabolites, which are the organic acids. So these were primarily represented by amino acids. And for functional implications, we group them according to the different pathways. So first we have the arginine metabolites, which were all decreased in the biosolids group. So these metabolites are known to play an important role in ovarian folliculogenesis, spermatogenesis, and also in thyroid development. So a decrease in these metabolites have implications in the fetal development of these pathways. So the next amino acid pathway that we looked at were the one-carbon metabolism pathway, where one-carbon metabolites like methionine, betaine, and pyridoxamine were all increased in the biosolids group. So studies have shown that increased presence of these metabolites could be detrimental to normal fetal growth and development. So the other pathway was the branch chain amino acid pathway, which we also saw to be implicated in the pathways result that we saw earlier. So valine, leucine, and alanine, which are part of this pathway, were all increased in the biosolids group. And increased BCAA amino acids is known to result in reduced fetal placental uptake of other essential amino acids, which could reduce nutrient supply for the fetal development. It's also to be noted that BCAA is also linked to lipid metabolism and fatty acid oxidation, which we saw to be significantly affected in the earlier pathway. Among the other class of metabolites was a benzene class of metabolites, which was elevated in the biosolids group. There were two important metabolites which are known to be markers of benzene exposure. So benzene is an important carcinogen, which is known to be inhaled and is known to lead to a lot of blood disorders primarily. So it's interesting that we saw two metabolites which was clear markers of benzene exposure in this study. So the network analysis here showed that the benzenoid class of metabolites interacted directly with lipids and amino acid metabolites, which could be implicating the observed phenotypic changes that we see. Apart from the endogenous metabolites, we also found about 40 exogenous metabolites, which are not a part of the normal metabolism of the animal and are introduced by external sources. So as you can see, most of these are sourced from drugs, food molecules, cosmetics, and pharmaceutical products. Interestingly, many of the differential metabolites that we saw in our study are also markers of inborn errors of metabolism. And two of them are in fact used in the clinical diagnosis of disorders of fatty acid beta oxidation. We also found two markers which are indicative of oxidative stress to be upregulated in the biosolids group of animals, which shows an altered embryonic metabolism and growth. So we identified some biomarkers of biosolids exposure by a combination of using the VIP plot, the t-test, and the S-plot. So on the right, you can see markers which are upregulated in the biosolid animals. And on the left, we have markers that are downregulated in the biosolids animals. So 15-HE3 which is known to modulate the arachidonic acid metabolism, which in turn upregulates the PPAR gamma pathway, which can lead to fetal programming. So pyroglutamic acid, which was downregulated in the biosolid animals, is an important intermediate in glutathione metabolism and hence plays an important role in ovarian development. So palmitic acid and oleamide were other important biomarkers that we discovered in our study. So looking at the fetal sex-specific differences. So I already indicated that these animals were from same-sex pregnancies. So we grouped the animals based on the sex of the fetus into animals carrying a male fetus and animals carrying a female fetus, and then looked at the the metabolite profile in these animals. So we can clearly see that animals carrying a female fetus had a more clear grouping of the metabolites based on the PCA and the OPSDA analysis, showing that animals carrying a female fetus had an increased response to biosolid exposure. So looking at the class level differences of the different metabolites based on the sex of the fetus, we noticed that the organic acids group of metabolites were decreased in the female animals carrying a female fetus as compared to animals carrying a male fetus in both the control and the biosolids group. Similarly, the benzenol class of metabolites showed a significant interaction between treatment and the fetal sex effects. So to summarize, so exposure to biosolids of the sheep via the biosolid treated pasture results in disruption of metabolites reflected in the maternal plasma, indicated by disruption of fatty acids, glycerophospholipids, amino acids, and nucleotides. And disruption of these metabolites functionally results in disrupted metabolic pathways involved in fatty acid oxidation and branch chain amino acid pathways, both of which are implicated in increased oxidative stress. And we propose that these combinedly contribute to the previously observed phenotypic changes in the biosolid exposed offspring, which are reflected by increased bone mineral density, disrupted reproduction axis, and increased thyroid weight. So some of the limitations of the study was that, as indicated earlier, we had a large number of unknown metabolites, and we also had an increased ratio of the sample to the metabolites, which was too high, which could have implications in the statistical analysis. Another limitation of this study was that the chemical composition and the dosage of the various endocrine disrupting chemicals and other chemicals in the biosolids have not been determined. So this could be lead to batch variance in the biosolids, but this is also reflective of the real life scenario where each individual is exposed to a different dose and different array of chemicals. So to conclude, the preconceptional and gestational exposure to biosolid results in alterations in the maternal metabolome, which could be contributing to the aberrant phenotypic changes, which has been noted earlier in this real-life EC exposure model. We also observed fetal sex-specific differences, primarily female-specific differences in the maternal metabolome in response to biosolid exposure, although the exact mechanism of this needs to be explored further. So this is the first proof of concept study to apply metabolomics to validate the effect of real-life environmental chemical exposure, reflecting the real-life scenario where every human is exposed to a different dose and cocktail of environmental chemicals. So it is important that we've used a translationally relevant sheep model, which has a developmental trajectory that is similar to humans. The results of this study are of great importance because biosolids are increasingly being used in agricultural, landscaping, and other uses. And crops grown in agricultural fields using biosolids have already reached the human food chain. So this study has far-reaching implications in the present scenario. Further details of this can be seen in the publication, which was recently published in Science of the Total Environment. So with that, I'm open for questions.
Okay, great job. Yeah, so this is very interesting research using very unique unique model. So while we are waiting for, so still I don't see any question yet. So while we are waiting for the question from the audience, so let me start asking a couple of questions. So when you design this biosolid study, and then so you are comparing the group, like the sheep, the intake this biosolid, like a grass grown from the biosolid one, and then another one is just organic one, right? So, right. And then
the control group was actually inorganic fertilizer group.
Inorganic fertilizer, okay. So what was your a priori hypothesis or expectation between these biosolids? So at the beginning, you mentioned that biosolid is a real-life mixture of this environmental chemical, but on the other hand, biosolid is a really good, like a nutrition standpoint, is a very nutrition-rich the fertilizer and soil, right. And then so it can impact and also impact this well-grown the grass, and then nutritionally also good for sheep and even human being. So at the beginning, so when you design this study, are you expecting that this biosolid group, in terms of this metabolic profile, and do you expect more disturbed direction, this biosolid group compared to the inorganic fertilizer? Can you talk about starting about this scenario?
Mm-hmm. Yes, there is speculation that in fact there are reports which show that the micronutrient content of inorganic fertilizer grown plants and that of organic grown plants, there is a difference, which could in turn affect the animal model as well. But we did not explore that part of the study because that would require us to characterize the grass from the two different scenarios to get an exact scenario of the micronutrient composition of the grass grown in control fields versus grass grown in the biosolid treated pastures field. But here the interesting part is that the exposure routes of the sheep to the biosolids is primarily via ingestion, and because it's consuming when it's consuming the grass grown on the biosolids field, it might also be ingesting some of the biosolids as well. And the other routes of exposure is inhalation and also absorption from the skin. So it's not only the micronutrient content of the plant which plays a role in the metabolomic differences that we see, but it's also from the other routes of exposure. So I think it's a comprehensive exposure, and it's the effects that we see is not only based on the micronutrient content of the grass.
Okay, that's interesting. So before you start this experiment, did you also measure any this nutritional profile between biosolid and then inorganic fertilizer? So nutrition, but also this endocrine disrupting chemical. So once we know what level of this, the chemical and nutrition, like input level, and then intake level, and then the metabolism, and then I believe that we can make a fair comparison between these two groups. And so I was wondering how you control the exposure level, external exposure from this biosolid versus fertilizer, and then and then when you measure this endogenous metabolite and exogenous metabolite, and then comparing those two.
Yeah, that'll be an interesting study, but that would be a separate study on its own because we'll have to do a mass spec for the control groups versus the biosolid groups. I believe our collaborators are working on getting the exact composition of the biosolids used in the study at that particular time, but we do not have those results yet. But we are also looking at other environmental chemicals, which was comparing between the control and the biosolid group, like PFAS is one chemical that we are seeing. So we looked at PFAS levels between the control animals and the biosolids animals. The analysis is still going on, so I cannot give you more details on that. But there are some interesting results there because some of the environmental chemicals the environment is so contaminated with them that it's not surprising that we see some of those chemicals in the control groups as well. But in terms of experimental design, the control and the biosolid pastures were physically separated with about a 1 kilometer difference between them. So there is no chance of cross-contamination between the two pastures and hence the animals.
Oh, okay. So because I'm not very well aware of the how the study was designed and the pasture and okay, 1 kilometer each other. Okay. Maybe I can ask one more question. So then you mentioned that at the end, just right before the question, so this question, and you mentioned that you detected the PFAS, and I think the PFAS is very interesting chemical recently detected in biosolid. So that's why that's one, the major external chemical that is of concern. But another important toxicant we are concerned from this biosolid is heavy metals. So lead and arsenic and cadmium. So I know you mentioned that this composition analysis is done in another group, but have you thought about this other potential contaminant we typically see in biosolid, and that in your study, like the potential impact of this heavy metal exposure, and then that made difference in this metabolic profile between those two groups?
So we couldn't get in our metabolic profile, we did not find any heavy metal compounds and things like that. But I believe there was a recent publication which has characterized biosolids based on a collaborative effort from multiple centers, and they have listed out all the chemicals that are typically present in a biosolid sample. So yes, so that does include a lot of heavy metals. But since we do not have the exact composition of the biosolids used for this particular study, we are not able to correlate the exact chemicals which could be contributing to the effects that we see. So what is the focus in our study is that we do see a lot of effects, which we are correlating with the established phenotypic model offsprings from this model. So we speculate that these specific metabolic differences could be contributing to the observed specific phenotypic changes from this model. So this study focuses more on the biological effects of the biosolids versus the other technical aspects like the composition or the relative effect contribution of each of the chemicals and things like that. But I think those are points of interest from the exposure assessment point of view, and that will be a great study if we can combine that effect as well with the biological effects that we see.
Okay. So we are still waiting for questions from the audience. If you have any other question, please put your question to the Q&A. I can see a raise, Marie. If you can unmute.
Yes.
Yeah.
Can you hear me? Can you hear me? Oh yeah. So I wanted to say thank you very much. This was a wonderful talk, and I'm going to ask a question that may be a little bit beyond the scope. I mean, I understand that this sheep model is really great for illustrating potential mechanisms relevant to humans, but I'm just thinking about the biosolids. It seems to me that there could be a kind of bigger picture environmental benefit of reusing human waste to fertilize crops. And I looked up the Easy Care U when you brought it up. And, you know, obviously these sheep are valuable for both wool as well as meat. And I'm wondering if you could comment specifically on, you know, the issue of use of biosolids as a way of reducing an environmental waste product. And I could imagine that even if there are some negative impacts of the EDCs in the biosolids, that for example, continuing to use this to feed sheep or other animals which might not be used for their meat, but just for wool, could be valuable. So it's kind of a bigger picture question, because you did talk about the implications of biosolid use for the environment. And I wonder if you could say something about that.
Yeah, there is no doubt that biosolids are a valuable source of recycling waste into use, because they are a rich source of nutrients. Like Dr. Sankyun Park said, it is very rich in micronutrients and could help in plant growth. But it's just that we have to be aware of what we are putting into the soil, because if the PFAS levels are as high as some of the reports indicate, then we might be damaging the soils, the composition, the micronutrient, the microbiome of the soil in itself, which could implicate further crop cycles, and which could in fact find its way into the human food chain. So I don't think in the current scenario we can completely avoid using biosolids, because that's one of a critical point of recycling of the waste in the waste recycling process. But I think it's important that we are aware, because a lot of people have been shifting to what we call as organic farming and organic crops in terms of even daily use. It's just that we have to be aware that even in terms of organic farming, if we are using biosolids, we do have associated risks that we need to be aware of. I think in the current scenario, there is so much chemicals all around us that it's impossible to avoid any of it, but we should make a personal effort to be conscious of what we use and what we put into the environment.
Yeah, thank you very much. Actually, Sung-Kyun, can I just ask one brief follow-up question?
Yeah, please go ahead.
Yeah. So I was just thinking, I think your point is really well taken, that there are chemicals and everything, and thinking about the PFAS as sort of an emerging chemical of concern. One approach that I'm aware of in real life is that you can actually use certain kinds of plants as a way of extracting certain harmful chemicals from the soil. And some of those could be metals or could be other types of contaminants. And since your sheep are based in the UK, I was just thinking about the Heathrow Airport in London as an example, where I had heard about them planting reeds near the runways to absorb some of the dangerous chemicals in the runoff, from jet fuel and things like that. So one thing that I think is worth mentioning is bioremediation, like kind of addressing that there are good nutrients, for example, in the biosolids, but there may be a way of, kind of minimizing what gets into the human body or the sheep, through selective use of plants. But I think that's probably a botany research question as well, but I'd happy to hear any comments on that question as well. Or maybe you know of other work being done by your collaborators in the UK.
That's an interesting thing. I'm not aware of them doing any bioremediation at the site of the where the animals are reared. But I think in the overall perspective, that'll be a good scenario, because we cannot really avoid escaping a lot of these chemicals. So we need to look at alternative ways of how we can prevent them from entering the system. And I think bioremediation is a good way to go.
Great, thank you again.
Okay, so we still have time, but if there are no more questions, then maybe we can end this seminar here. So thank you, Viveka, for your this excellent presentation and a very interesting topic. And I think we can have another chance to talk more about this biosolid issue. And as Marie mentioned, that maybe we also have to think about another like aspect of how this biosolid issue can be handled, not just this environmental chemical source of environmental exposure, but also how to prevent and then reduce this waste product, and then remediation and many other things. And then we will need to think about the role of this biosolid in many different aspects. So thank you for your presentation again. And then for the audience, thank you for attending this meeting. Yeah. So we will see you, actually we will see you during February, so January and then February, EHS has many the faculty candidate seminar. So please attend that seminar, and then we will see you in March. Bye.